Oysters and mussels have been in our oceans for over 145 million years. Blue mussels are native to European waters, while the Pacific oyster was introduced by humans in the 1960s. Their role is important, helping to filter nutrients, remove pollutants, and provide a home for sessile fauna, which are the non-moving animals that have mastered life without mobility by adapting to rely on ocean currents for nourishment and reproduction. Two marine biologists from the Toronto Marine Laboratory in Sweden are exploring the intricate life that flourishes on these outstanding species.
So we're in Tiarno here today and we're looking at the oysters and blue mussels in 4 different locations, and our aim is to look at the difference in biodiversity of the sessile fauna on mussels versus oysters and also across the 4 different locations. It's important to look at these because they are foundation species for the ecosystems, both here at the port and piers and also in silty areas. So for the experiment, the team's gonna go out to the 4 different locations, 2 man-made, 2 silty, and we're gonna go snorkeling. We're gonna collect mussels and oysters from all 4 locations and then bring them back to the lab, analyze the sessile fauna that is growing on both the oysters and mussels, and then measure biodiversity from that.
We are here in Toronto Marine Lab getting ready to analyze our samples. We have them living in a flow tank between the time that we analyze and put them back into the waters. We will be using microscopes to identify the sessile communities that are present. This is an example of an oyster from our man-made sites. You can see that there is a whole new planet living on top of this shell. Without this shell, these bivalves would have to find another substrate to live on. These are bivalves. You could see that there's a mix of other sessile taxa that are living on it, and we will be using the microscopes to figure out just which ones are using this. We will be using an image software to calculate the percent coverage of each taxa on the different sides of the bivalves.
Through analysis, our dedicated researchers unveiled 3 differences. First, a distinct contrast emerges between habitat types and percentage cover, highlighting that both abundance and diversity of sessile fauna vary across different habitat locations. Additionally, a statistical difference was identified between taxonomic richness and habitat type. And finally, a differentiation occurred between the percent coverage of unique taxa per bivalve. This suggests that the area of each individual taxa is shaped by the specific characteristics of the bivalve. The experiment contributed another piece to the puzzle, shedding light on the role of oysters and mussels as foundational species.